To the naked eye, acute inflammation appears as simple swelling and redness, but at the microscopic level, it is a highly orchestrated, algorithmic biological response driven entirely by chemical mediators. To understand how the human body regulates vascular permeability, registers pain, and coordinates an immune army, we have to divide these chemical signals based on exactly where they originate. The first major class consists of cell-derived mediators. These are chemical signals synthesized and secreted locally by tissue-dwelling cells, like mast cells, right at the exact site of injury. The second class consists of plasma-derived mediators. Instead of being manufactured on site, these are systemic proenzymes that constantly circulate in the bloodstream, waiting for a specific biological trigger to set off a chain reaction. Mapping the acute inflammatory response requires tracking both axes simultaneously, observing how localized cell membrane breakdowns sound the immediate alarm, while circulating plasma proteins flood the zone with systemic munitions. Even within the local tissues, cell-derived mediators operate on two distinct timelines. The physiological response begins with pre-formed mediators. These are chemicals, specifically histamine and serotonin, that are already fully manufactured and stored inside granules within local mast cells and circulating platelets. Because they are pre-packaged, there is no delay for protein synthesis. The moment tissue injury occurs, these granules are released instantly into the surrounding environment. The physiological result is immediate. The sudden spike in local histamine forces the capillary walls to rapidly dilate and become highly permeable. By instantly altering the structural landscape of the local blood vessels, these biological first responders prime the tissue for the slower, more complex biochemical cascades that follow. Once the initial alarm sounds, the second timeline begins. This phase relies on newly synthesized mediators, which are manufactured on demand primarily through the arachidonic acid pathway. The raw material for this pathway comes from the host cell itself. An enzyme called phospholipase A2 cleaves the structural phospholipids of the cell's own membrane, freeing a 20-carbon fatty acid called arachidonic acid. This free arachidonic acid serves as a master precursor. It acts as a biochemical fulcrum, immediately diverging down one of two distinct enzymatic pathways. The first branch is driven by the cyclooxygenase, or COX enzyme. COX converts the precursor into prostaglandins and thromboxanes. This specific branch generates the direct symptoms of an infection. Prostaglandin E2, for instance, heavily sensitizes local nerve endings to mediate pain and signals the brain to trigger a fever. The second branch is driven by the lipoxygenase, or LOX enzyme. This converts the arachidonic acid into a different class of mediators called leukotrienes. Rather than triggering pain, the LOX pathway handles logistics. Leukotriene B4 acts as a highly potent chemoattractant, laying down a chemical gradient that pulls circulating neutrophils directly to the site of the injury. In this phase, the host cell's own structural membrane is literally cannibalized to generate the precise chemical signals required to orchestrate local pain, fever, and immune recruitment. Because this pathway is highly predictable, modern clinical pharmacology can suppress inflammation by directly targeting and blockading specific enzymes within the arachidonic acid tree. Corticosteroids operate at the absolute root. By inhibiting phospholipase A2, they completely shut down the initial synthesis of arachidonic acid, silencing both branches of the inflammatory response. Non-steroidal anti-inflammatory drugs, like ibuprofen, operate further downstream. They selectively inhibit only the COX enzymes. By reverse engineering these pathways, physicians precisely amputate specific symptoms of the local response. A patient can take an NSAID to eliminate the pain and fever generated by the COX pathway, without shutting down the immune system's ability to recruit white blood cells via the LOX pathway. While local cells process their own membranes, a second front operates in the bloodstream via the plasma-derived mediators. The most critical of these is the complement system. This is a network of inactive proteins, labeled C1 through C9, which are synthesized by the liver and circulate continuously. The complement system does not synthesize new molecules. It relies on a cascading chain reaction of proteolytic cleavage, where one activated protein physically slices the next protein in the sequence into active fragments. The initiation of what is known as the classical pathway begins the moment an IgG or IgM antibody physically docks onto a bacterial antigen. Once docked, the antibody activates complement protein C1, triggering a sequence of cleavages that assemble molecular engines on the pathogen surface to form C5 convertase. Simultaneously, C3b fragments coat the bacteria for phagocytosis, while C5a fragments drift away, serving as a systemic chemoattractant. The complement cascade acts as a circulating tripwire, converting a single localized antibody detection into an exponentially amplifying signal that paints the target for systemic destruction. At the very end of this plasma-derived cascade, the complement system generates its ultimate lethal physiological payload, the membrane attack complex, or MAC. The MAC assembles from the final cleaved components of the cascade. Subunit C5b locks together with C6, C7, and C8, followed by multiple copies of C9. These combined subunits form a rigid, highly stable, cylindrical molecular drill. This complex forcefully embeds itself into the lipid bilayer of the target pathogen, punching a permanent hole entirely through the cell wall. This puncture destroys the pathogen's osmotic regulation. With the barrier breached, a massive, uncontrollable influx of water rushes inside the cell. The bacterial cell swells rapidly until its structural integrity fails, resulting in violent osmotic lysis and immediate pathogen death. Acute inflammation relies on a precise spatio-temporal duality. The localized, cell-derived breakdown of host membranes sounds the chemical alarm, while circulating plasma-derived complement proteins assemble into molecular drills to physically execute the invader.